Okay, so we are recording. So this is the Neuroscience and Philosophy Salon. So today we're really excited to have Lucina Uddin present to us her views developed over a couple of decades on the ontology and the existence and the understanding of large-scale functional brain networks. So Lucina is going to present for around half an hour, 35 minutes, and then hopefully we'll have a lot of discussion. So Lucina is a professor at UCLA in the US. So Lucina, please.
Thanks, Luis. It's always fun to get to talk about one of my favorite topics here with such a great crowd. Thanks for showing up. So these are kind of thoughts, works in progress, also meant to be controversial. So I hope we'll have some good discussions. You can see the screen okay? All right. So, as many of you know who work in cognitive neuroscience, we've been spending a lot of time thinking about spontaneous brain activity over the last 30 years because you can see there's actually a lot of structure to the kind of things that the brain does when we're at rest. So, it's not just kind of random noise. It seems to be in these low frequency fluctuations. When you put someone in the scanner and you just ask them to do nothing, you can actually see the coming and going, the formation of networks that look like they would be involved in visual processing or motor processing or memory or attention or language. So this kind of resting state fMRI or functional connectivity has given rise to a whole new world, I guess, of human connectomics research and network neuroscience. And a lot of that sort of methodological advancement and movement in the field is driving some of the conceptual issues that I'll be talking about today. So we all kind of know these things are happening, and we've started to call what we see as these, coherent fluctuations. We started to call them large-scale brain networks or functional networks. And I wanted to pause for a moment and think about where we're going in terms of that, explaining these phenomena and having the words to describe them and talk about them in the field. So this is just kind of an overview of the last 30 years of when, the first paper was published showing that you could find this coherent, oscillations across motor cortices, for example, even when the participant is not doing a motor task. And that after that followed years of discovery of networks. The default mode hypothesis of brain function was published in 2001. And then, these large data sharing initiatives began with the Human Connectome Project, and there's a lot of other ones now that are ongoing, looking at brain development. People are even using this method to look at fetal brain. You can look at brain before birth. And so with that, we've come across the need to kind of solidify some best practices for data analysis and sharing. And so a lot of societies, like, for example, the Organization for Human Brain Mapping, puts some effort into establishing best practices. And this has kind of led to an explosion of research in this field. As many of you are probably involved in the research are aware, it's hard to stay on top of it. And I'm going to argue that even more so, it's hard to stay on top of it because we haven't converged on some of the key issues regarding nomenclature and language that we use to describe large-scale functional brain networks. Just to give you an example, in other fields in science, we try to come up with taxonomies for describing and classifying information in a way that helps us basically organize what we've learned and communicate across fields. It's kind of a logical thing to do, to come up with some standard nomenclature so that when folks enter the field or they want to talk to other labs, we're speaking the same language. That's the goal. So in cognitive neuroscience, in the field we're talking about now, like human connectomics, we inherited a lot of the language from cognitive neuroscience, which of course inherited a lot of the language from psychology. If we really want to go back to it, inherited from philosophy. But we've done some work using kind of meta-analytic approaches to look for big picture themes in the corpus of cognitive neuroscience literature. So Taylor Bolt in the lab a number of years ago did some exploratory bifactor analysis applied to task fMRI contrast maps from Human Connectome Project task data, NeuroVault data, and just kind of found that there was a general factor that explained about half of the variance in task activation maps across many, many different kinds of paradigms. And this factor, if you kind of look at it closely, It resembles what Fox and Franson and others found in resting state fMRI data now 20 years ago, that you have this kind of general antagonistic relationship between what people call the default mode network here in blue and the frontal parietal task positive attention networks over in orange. So this kind of resting network organization is mirrored in what you see in the task literature. There's, of course, in this bifactor analysis, he found that there's other subtypes within this general broad task negative, task positive systems that also explains additional 31% of the variance. And what I would argue is that the problems we inherited from cognitive neuroscience are the way we talk about some of these systems is really almost lab dependent in some ways. So some folks will call these positive systems a multiple demand network, or others will call it domain general. I've heard it described as an extrinsic mode of function. And I think the most probably widely used term is executive control or executive network, or for these frontal parietal system. But across the field, a lot of these same regions are talked about using very different nomenclatures. The default mode network, when it was named, that name kind of stuck. And so these regions that are really in the medial prefrontal cortex, posterior cingulate, that name seems to be pretty stable and widely used to describe this certain set of brain regions. I mean, the question I'm going to ask is really like, is this important? Like, what's in a name? Does it matter what we call these things? And I'll argue that, well, if anything, if for no other reason than it's hard to communicate if we don't. And I'm going to give you this example of a few descriptions of large-scale brain networks that involve some of the same brain areas. So prefrontal cortex, insula, dorsal anterior cingulate. In one paper, that's referred to as a cingulo-opercular network. In some of our work, we've called the same set of regions the salience network. And if you dig back even further, it's often some of these same areas, brain areas, come up in the when you talk about the dorsal and ventral attention networks. In particular, ventral attention network seems to share some of these same brain regions. So there's a lot of overlap in terms of anatomy here. Is it the same? Is it different? What are we talking about? And to me, this is particularly shocking, I think, if you look at some of the atlases that people use to describe these networks and give them names. So if you look across 4 widely used brain parcellation atlases, there's something called a salience network. In some of these atlases, it's called the salience/ventral attention network. In some, they're broken into anterior and posterior. But if you can see with your eyes, you don't even need to quantify that these are not the same sets of brain regions, even though people seem to be calling them the same thing. If you're a stickler for anatomy, you might be, worried that we're going down this route. I've thought a lot about this, what people are calling. Some people are calling the salience network, and this is where my lab does a lot of work. So I find it particularly distressing that we haven't converged not only on this name, but on many of the names that I'll be talking about today. We do a lot of work in the lab looking at interactions between networks, and we have a theory about how the salience or singulo-insular systems, facilitate switching between the default mode and lateral frontal parietal or executive networks. As sort of what we do in the lab, on the science end of things. And this example of the insular cortex, I think, illustrates how hard it is to name networks, in particular with regions like this area, anterior insula and the anterior cingulate. They're just activated across a lot of different cognitive paradigms. It's a functionally flexible part of the brain. So this is some work from Thomas Yeo about 10 years ago, using a hierarchical Bayesian modeling approach applied to contrast from the brain map database, and just showing that like across very many different types of cognitive paradigms, these regions pop up a lot. So it's kind of a functionally very flexible brain region. This is the stuff we did with Luis again about 10 years ago now, also thinking about the diversity of the tasks that are activated when you see insula activation. It's a very functionally diverse brain region. So it sort of makes sense that it might pop up in a lot of these different network configurations. And there's reasons to believe that might be because it's sort of dynamically affiliated, to use Luis's terminology, dynamically affiliated with different brain areas. And you can use approaches like sliding window functional brain dynamics. And I won't get into methods today, but we can talk later. But kind of these kinds of approaches show you that, there's different ways that the insula interacts with the rest of the brain. So some work from Jason Nomi in our lab a few years ago, showing that the insula kind of, especially the dorsal anterior insula component, kind of changes its network affiliations from moment to moment. So, if you look at these dynamics on a fine scale, you can realize that it's kind of talking to lots of different brain areas over time. So I'm just going to get into why I think, getting to the root of these naming issues is going to be really important for translational work, psychiatry in particular. This is from Tom Insel and Bruce Cuthbert's paper a while ago when they were first talking about the RDoC, or research domain criteria. And they just kind of put insula here in their diagram of a brain area that might be interesting to study in a transdiagnostic way. And I completely agree. I think that insula is implicated across things ranging, clinical conditions ranging from depression to anxiety to addiction to schizophrenia, frontotemporal dementia. It seems to be one of those areas of the brain where if you understand what's going on there functionally, it can help you understand, give you clues into dysfunction across a lot of clinical domains. So I think it's important, not just for us interested philosophically in what do we call what, but also for the translation to the bedside, essentially. If you look across the literature, I'm really amazed at how much these areas, the insula and ACC, pop up in these studies of mental illness. So it's like, seems to be a common neural substrate across, and this is structural MRI studies from a while ago showing, common gray matter volume reductions across a range of psychiatric conditions. In a nice recent paper in Nature, Charles Lynch and colleagues showed that the salience network insula ACC seems to be expanded in the individuals with depression. It's kind of larger. And so I think that is important, if we're going to talk about as a marker of treatment response or as a biomarker for some disorder, really being more precise about what is the salience network might be a good place to start. And then if you look at across addiction, now that I'm working a lot with the adolescent brain cognitive development study, and it's a long-term longitudinal study looking at child mental health and development of substance use disorder. And again, like the insula and ACC seem to be important for alcohol, nicotine, use disorder risks, and all of these kinds of things related to addiction. So really just important circuitry for us to get a handle on. So this leads us to the problem, as I see it. How do we define these functional brain networks, and can we ever get to a standardization of network nomenclature, which I think would have a significant impact on clinical translation? We started this was Nathan Sprang and I at a conference called the Alpine Brain Imaging, ABIM, Alpine Brain Imaging meeting a few years back. And we were taking a break from skiing, and we thought maybe we can just write a paper on this topic, while we're here in this, sitting by the fire. And this was kind of the first pass that we thought, oh, how hard can it be? We'll just come up with a taxonomy. We'll, give these networks some labels, and people will go with that from now on. And we brought in our friend Thomas Yeo because he's created a lot of these brain parcellations that people use, and he's one of the, he's used. I think he's probably created the parcellation that's the most widely used in the field. So the names that he put on the networks are often the names you'll see in papers. So we thought for our first pass, let's go ahead and try to move towards an anatomical nomenclature that has nothing to do with function, because function, we keep discovering it and things can change, and people have different theories as to what functional significance of some brain network might be. But with anatomy, it's harder to debate. So we thought, let's just go with we'll look across various networks. You've got like Thomas Yo's up on the top. You've got Evan Gordon, another popular parcellation scheme, on the bottom. Most of them have a visual network somewhere in the back of the brain. It might be one or two or three, but we decided that if we're going to go with an anatomical nomenclature, we would recommend to use the phrase occipital network. Oops, I went the wrong way. And similarly for like the motor cortex, if you're going to talk about our somatomotor or sensory motor, most of these parcellation schemes you see in blue on top, and you see even further broken down motor and sensory cortices on the bottom. We can safely, I think, say that that's pericentral network or pericentral in terms of the regions that we're talking about. And then you have things like dorsal frontal parietal, which often goes by the name of dorsal attention. But if we want to just say, okay, we're going to be agnostic about what it does, but we know it's somewhere in the dorsal and somewhere in the frontoparietal cortex, we can get a little more anatomically oriented. And similarly, we said, let's forget about executive control for now and just call it lateral frontoparietal, just for the sake of consistency and, trying to get some consensus. And then this was the tough one, the cingulo-insular salience, ventral tension. It's just so many brain regions seem to pop up again and again across these three different nomenclatures. And in fact, in the Gordon parcellation, he's labeled them as three separate networks, where in Yo, he's put them together. Very confusing, I would say, when we're trying to do things like a literature search. So let's just forget it and call it mid-cingulo-insular. That was our suggestion. And then we have to go forward. Okay. And then this was the default mode. And, everyone likes that name. So I don't think this suggestion is going to stick, but it is really just a medial frontoparietal network. And if we want to stick with the nomenclature that's purely anatomical, that would be our suggestion. So this was a paper we put out there about 6 years ago now, just saying, here's one way to think about these networks that we're all using in our work, and we're all trying to get a sense of what they do and how they behave. Maybe for now, it would make our life easier if we stuck to some anatomical terminology, at least so that we can get on the same page. And what ended up happening was, well, this didn't work. And in terms of nobody wanted to adopt these new names. Everyone's been doing what they're doing. Nobody was interested in changing their default mode network to the medial prefrontal network. There's a lot of things we left out, right? We didn't talk about subcortical brain structures, individual variability, the fact that you can break down these networks further and further as you go, into increasing model orders, for example, with the independent component analysis. We kind of took the first pass, and it was a gross oversimplification. But I would argue, frankly, that everything that we do in networks neuroscience in this large scale, macro scale level is a gross oversimplification, because even in the 90s, before we started like heavily using fMRI, there was this idea that cognition is served by interconnected neural networks, complex behaviors mapped at the level of multifocal neural systems rather than specific sites. It's not sort of a one network does one function. We've known that. Work from Randy McIntosh and others for well over 20 years have talked about the importance of neural context for understanding the functional relevance of a given brain area, and understanding the status of the rest of the brain is important for function of one area. And Luis, has a great paper in 2014 talking about how the network needs to be understood in terms of interactions between brain regions as they unfold temporally. So I think we've always kind of pretended that we could delineate networks in this very fixed way, when we're really looking at a moving target. Networks come and go, I guess. They fluctuate. This is work from Taylor Bolt in my lab a few years ago, and he found that there's basically three main spatiotemporal patterns that you can kind of chart over time that account for a lot of the observations in the literature, like the default mode anti-correlation with task positive, time lag propagation literature, gradients, functional connectivity gradients. You can capture a lot of these phenomena in just three main spatiotemporal patterns. And we're getting, maybe the networks that we're talking about are really just snapshots of this kind of dynamic, oscillation. And I think we sort of tend to just oversimplify when we say, okay, there's these 6 networks or 5 networks, these snapshots. And so what we tried to do was form this best practices committee through the Organization for Human Brain Mapping. They have a long history of doing this. The Committee on Best Practices in Data Analysis and Sharing was chaired by Tom Nichols many years ago. And there's a committee on MEG and EEG best practices. So this one we called Anastasia Indiki called this the Work Group for Harmonized Taxonomy of Networks, the WhatNet. And we spend a lot of time with this group. A bunch of folks on this author list are creators of those brain parcellation tools. They've spent a lot of time doing cross-species work. Some of them do EEG, MEG. Some are computational modelers. Luis is on there. We've got a philosopher. So I would say we spend about 6 weeks talking about what is a network. Is there such a thing as a network? For all the reasons that I just mentioned. We don't even know if this is an entity that's worth giving that that name to. But we finally decided, well, people are calling things networks and they're pretending that there are networks. So we, at the very least, we have to go with what's out there right now. So a lot of meetings, a lot of discussions that culminated in this perspective piece. When was that? 2023? I don't know. Seems like forever ago. And we did, throughout the course of that best practices committee, we put a survey to the community and we said, okay, if you saw this in your lab, what would you call it? Just name the network. Free response. Most people said default mode network. Okay, great. This one, which as I mentioned, is more controversial, got a very little consensus. So we had, 100 or so respondents. No, we had several hundred respondents. But the things like the default network or motor network were non-controversial in terms of the naming, but things like salience, frontoparietal, things that were more association cortex based, where really there was very little consensus across people when you ask them, what do you call this? So just it really illustrating the problem out in the wild that we don't agree on what these things are. So we tried to get some minimal reporting guidelines. The best we can do is say, hey, if you're writing a paper, you're doing a study, try to follow some of these conventions in your methods and results, similar to what the original best practices committee on data analysis and sharing were doing. I said maybe we should consider the anatomical labels. If you're going to use the functional labels, fine, but please also try to add some anatomical labels and try to use a particular, mapping to one or more parcellation schemes to help comparison across groups. And so this was endorsed by the OHBM, and we thought, okay, we have done our job, we can move on with our lives. But, all of these, these processes kind of led us to the conclusion that it's harder to do this than one would think. If you have a grad student who comes into your lab, they show you a map and they say, look, I found this, and depending on who you are, you might say, great, we're going to write a paper about the salience network. We're going to just get into that literature and that's the paper and here we go. If you're someone else, you might say, okay, great, we're going to talk about the ventral attention network and here's the ventral attention network literature and we're going to go that direction. And if you're from a different lab, you'll say, well, that's clearly the cinguloopercular network. In fact, now we're calling it the action mode network. A lot of times people change their mind about what they're going to call things. And I think like for a grad or for anybody really entering the field, it's just not, we just don't have good reporting guidelines or even good ways of deciding how to contextualize our findings in the broader literature. And so that's a missed opportunity. And I think it's even a greater missed opportunity for clinical science when people are unaware of these issues in the field, especially people new to the field. So we put together this network correspondence toolbox. Ruby Kong and Thomas Yo's lab put together this lovely Python tool, just came out earlier this year. You basically, we've included in it something like 26 different parcellation schemes, including the most commonly used ones, Yo, Gordon, Schaefer, Shen, and many others from ICA. And it's actually a toolbox in which you can upload your own atlas if you desire. And what it does is you can take your data file, whatever you've got, stick it into the atlas and say, I want to compare my results to these 10 atlases or 5 atlases or however many you want. It's super simple. It just computes the dice overlap between whatever you've input and all the networks in all the atlases. It spits out some, I love this. Okay, it spits out some quantification of the overlap, runs a few statistics so you can say, well, whatever I have, it overlaps a lot with what Yo is calling dorsal attention. And then what I love about it is that you can play around with it and you can say, I'm going to put in Yo's atlas and Gordon's atlas and look at how their nomenclature corresponds to each other. And to me, this is really revealing. So if you look on the y-axis here, this is Yo's labeling. What he calls salience ventral attention B overlaps very much with what Gordon calls cinguloopercular and also salience. So, it's almost like his salience attention B is a combination of Gordon's cinguloopercular and salience. And I think to me, this is like a nice way of like making sense of the world of, and it spits out some nice diagrams. So you can, it gives you like how much overlap you have with each of these different networks, and, some whatever p-values and things if you want to make more quantitative claims. It's pretty user-friendly. This is just an example. If you take the human connectome project data, the working memory task, 2-back versus 0-back, it's got some activation you see there in yellow. If you threw that into our toolbox, it would tell you, and you said, like, compare it to these 8 atlases on the bottom, it would tell you, well, it overlaps a lot with what Gordon calls dorsal attention frontoparietal, and you could see that this one's pretty good in that there's a lot of overlap with what different people are calling frontoparietal/dorsal attention. This one is more tricky. This is kind of just a messy map of some ICA, random ICA component. And it's messy, but if you throw it into the toolbox, it can give you some like best fit with the different atlases that you input. I'm just going to wrap up here, but there's a transmitter article about this. They called it a decoder tool, and I kind of love that. It translates your findings and other people's findings in terms of different brain parcellation atlases. Mind you, like, we haven't solved any of the problems I talked about. we haven't dealt with the nomenclature issue and the differences across it. All we're trying to do is make it easier for people to translate their findings and find out how much they, correspond to other people's findings. But we have not gotten anywhere near the problem of, like, do networks exist, and are they dynamic or static, and what do they actually do, and what are their functional neighbors? We have made zero progress on that particular set of questions. What we have done is tried to make practical tools for practical people, but I'm very much looking forward to discussions about the actual issues that I brought up. There's I gave this talk at OHBM last year as a keynote, and it's actually online if you want to see it. It's more on the science and less on the philosophy, I guess. But the idea was that we're trying to get towards standardization, why, these network definitions matter. We're trying to get people or help people to be more quantitative in some of these comparisons, provide stats, tools to do the stats, look at cross-atlas comparisons, user-friendly design. And really, I'm really primarily concerned with how, when we as neuroscientists aren't clear with our language, how that negatively affects the downstream translation to psychiatry and neurology, where they're, I think, looking to us to say, what is this? What are we seeing? And if we're not clear about it, there's no way that we can hope to make progress on, look at some of these disorders. So I think as, the field matures, it'll be really increasingly imperative to arrive, if not a consensus, at least a way of reporting our results and studies that will facilitate, discovery. And this network nomenclature, I think, is one of our biggest hurdles to overcome. The toolbox I mentioned, the network correspondence toolbox, will help us in the short term to translate findings across labs while we work towards this standard creation and adoption of a universal taxonomy. This is the group, or a lot of the group, who was involved in these discussions and whatnot and creating the toolbox. We like to meet up at OHBM when we go. This was a Glasgow meeting. This is a paper from me and Bharat this year. Even though, like, nobody sort of is calling things with these anatomical labels, I think we maybe that's one of the first steps is just to make sure we're clear about what parts of the brain we're talking about when we call something a particular network. And I don't think we're there yet in terms of this taxonomy problem. I don't know what it would take to get us there. But these are sort of the processes we've tried. Don't say we haven't tried. But this is where we are at this moment in time. So I'm happy to stop there so we have plenty of time for discussion, and thank all the individuals who are really involved in this from the start. And thanks, Luis, for giving me a soapbox.
Oh yeah, thank you so much. That was really wonderful. Yeah, so I think that we have a lot of people today. So I'll try to keep track of your hands if you want to participate. This is for everyone, for us to discuss all levels. So if you have a question, please raise your hand and we'll try to keep track. Damaris, please go ahead.
Thank you so much. This was very enlightening. I'm not from the field, and that clears up a lot of the confusion I had. I think what
Great.
I'm wondering about is, I mean, besides historical reasons that you mentioned, like people like the names, and the approaches,
Mhm.
but like how much worth would it be to look at it from a computational angle? So to be like, okay, well, this network has a lot of recurrent types
Mhm.
of the cell, and it has these type of long connections, and this
Mhm.
one does not. Like,
Yeah.
how could one use that for distinguishing regions?
I think that's exactly, part of what needs to happen. A lot more of this kind of quantitative, I would say like head-to-head comparison, right? Like use whatever the metrics are to say, okay, this actually does have more similar, whatever it is, cell types or things like that. We've been sort of focused almost exclusively on functional, magnetic resonance imaging, fMRI data, and that's part of the problem is, we've and now, like I said, we inherited whatever problems cognitive neuroscience had from the beginning, and which we can blame psychology if we really want to blame somebody. Um, I know Luis has thoughts about this, but because we think there's such a thing as attention, or we think there's such a thing as whatever, and then we see something in the brain that lights up when we do an attention task, we say that's the attention network. And so we all do this, I think we're all sort of guilty of it, rather than let's find out what are the range of functions that actually are, I think we tend to impose our own kind of interpretation on something. And so I do think we have to do a lot more meta-analytic type of work, and a lot more discussion with folks outside of the fMRI bubble, I guess it is, where we look at everything through a particular lens. And I do think that I would love for people like you outside of this field to contribute to this discussion. So thanks.
Yeah, just a quick follow-up. Like, do you think like are the brain regions distinct enough from the type of cells that they have
Oh,
and their recurrency and their connections,
yeah.
like at least the length of connections, that might even be a possibility?
Well, so using fMRI, a lot of these brain parcellations are created in that way. So they're like this network has a specific pattern of connectivity, and that's why we call it that network, whereas this one has a little bit different. But those are all connectivity, which is statistical correlations between brain regions from the fMRI signal. So it's not sort of not necessarily anatomical
Mm.
connectivity, but then sometimes you have diffusion imaging, and then you get like anatomical connections, and you can do parcellation based on that. So the problem is like you can do parcellation based on cytoarchitectonics. You can do it based on myelin. You can do it based on all these different metrics, and you'll get slightly different networks depending on how, or even receptor density, right? So genetic markers and things like that. So depending on what you use, you'll get a different brain parcellation. And so that's also part of the problem.
Yeah. Okay.
Mhm.
Thank you.
Mhm.
Okay, Ellen, you want to go ask your question, please?
Sure. Thank you for the talk. I've been doing some salience network stuff for the past few years and also inter-individual variability stuff. So I'm very interested in your work and also the difficulty of different atlases calling different things, especially with respect to the salience network. I wonder to what extent any of these goals are possible to address, though, because of inter-individual variability in large part. Like
Mhm.
group atlases make the assumption that networks have the same spatial layout across people, and also often that there's no overlap between networks. And like you're saying with the salience
Yeah.
network, like the insula does a lot of different stuff.
Mhm.
It's probably a hub in some meaningful sense. And then there's also, definitely other hubs in the brain.
Mhm.
And so even saying like this region belongs to this network opposed to like
Mhm.
five networks,
Right.
is just not, it's not true.
Yeah.
And so I feel like to some extent, like we'll never have a taxonomy because
Right.
we can't because of the inter-individual variability and
Yeah.
maybe, dynamic changes over time,
Yeah.
and across potentially cognitive states, though most of it seems to be stable across cognitive states.
Right.
So like I get, I have on a very personal level feel like the pain of trying to compare across studies because of the
Yeah.
labels. I'm like, when I first started this, I'm like, oh my God, there's like 7 different salience networks. But
Exactly.
like maybe the solution is just to stop with this tool that Thomas Yo's group came up with. So we like know how similar what we're looking at is to all these different atlases to try and communicate with each other.
Yeah. Yeah, I mean, I totally agree. It's kind of what conclusion we came to. We can't stop people from doing this, but we can make it easier for you to compare what you're doing with the rest of the literature. I think what you said about inter-individual variability is certainly a concern, and there's a lot of folks who've done a lot of great work showing that, you know, networks can have different spatial topographies depending on the individual. I think what's heartening though is that even like you can find a default mode network in everyone. I've never seen a data set where you can't identify it, or you know, there's some things that just like seem to be, you can find pieces of these networks in hemispherectomy patients where there's like one entire hemisphere removed, and the remaining hemisphere you can sort of, if you use the, you know, toolbox, you can compare, and you, so it's like it's and you can find some pieces of them in a fetal brain, you know, so it's interesting that despite the individual variability, there are some consistent things that pop up that suggest that there's like some overall, I mean, there's going to be deviations from it, but there seems to be some like overall structure. But those things we're calling networks are still, they're approximations. They're like a static snapshot. They're maybe on average, you'll see the brain do that, but they clearly jump out of these formations for different reasons, right? So, but the problem is the field has already used this terminology for so long that it's hard to like, how do we get people to stop? Could just stop doing it, but you know, that's not going to happen. So I think like it was more of just like, we give up and we will provide tools for you to, you know, to make sense of the literature, but it's hard to tell people what to do.
As a kind of follow-up to that, I'm a clinical psychologist who does neuroscience research, and to maybe defend psychology a little bit, I think there is a lot of utility to having names that tap into at least some of the function of a brain network or region, because it helps people make sense of what they're reading, and like you know, just from a memory formation retention perspective, having something to anchor it to can be useful.
Yeah.
So yeah, but there's trade-offs then, because then people will make the assumption that these regions like only do this or something like that.
No, that's it. You've said it exactly right. And I think that's the philosophy problem here. It's like, it's not that it's not useful to call these networks by functional names. Is that have we gotten to the heart of what they are? You know, have we really like done this correctly? Maybe it's useful and will continue to be useful, and we're just going to have to live with the fact that we are missing some pieces of the full picture. But yeah, I totally agree.
Okay, great. Actually, I wanted to squeeze in here and ask you a question, because one thing that I'm really find more and more fascinating is about the dynamics of networks. So, and I wanted you to kind of, as an insider, you can tell me where I'm wrong in the following sense about the community. It's not like a tricky question. Yeah. So when I talk to people or I go places and hear things, networks are pretty much universally known, and people work with them as part of their vocabulary in the field, right? So, but one thing that is interesting is that the part that about dynamics, and that's the part that I wanted a little bit of feedback, seems to be less understood or appreciated more broadly. So the interesting thing is that more recently, we just submitted a paper, and the first time I did some kind of analysis on resting state data myself, and we were interested in dynamics. And so we did a lot of things, and I was quite ignorant of the field, and we did a lot of things. And then I started reading and seeing that this world of dynamics is huge. Like a lot of people have done so much. So I was like, oh, our results are so great, this and that. And everything had been done already. So in a sense, right? Because there's so much that has been done, and going way more than 10 years. And it's not like in the last 3 years. So here's my question. First, the more pragmatic, or the more kind of your view of the field. Is it true that the dynamics part hasn't caught on yet more broadly in the community? Obviously, there's this ton of work that I was kind of like, oh my God, it's all
so
over the place.
If you go to OHBM, it's definitely caught on. It's been since well over 15 years at OHBM. We talk about dynamics all the time.
Right,
but I think in that community, it's very well integrated. It might not be in like Cognitive Neuroscience Society, or well, you know what I mean? So it depends on where you go.
Yeah, but so if you think of I mean, I'm sorry to say, but like an average typical cognitive neuroscientist or a neuroimager. I mean, whatever that means, because we're all completely different. But
Yeah.
it seems to me that when people talk about it, they don't even so people incorporate in their way of thinking
Mhm.
networks,
Yeah.
but they don't seem to incorporate the fact that they may or may not be, depending, where you go and you study. Although the evidence is pretty clear that they are dynamic, so
Yeah.
I should make that clear.
Yeah.
It hasn't seemed to caught on, but I could be wrong. So here's a little bit more difficult question, which is, do you think that, so my reading of this literature and a little bit of what we found too, is that you see a lot of mixing of the standard networks. So it's not a replication of a one-to-one. In the dynamics is not that dynamically I'm in one network, I switch to another and I transition to another. It seems to be these hybrid things, not only during the movement, but also a little bit more stably. So they're not just uh, snapshots that one picks up and just uses in an ad hoc manner. It seems to be that some of these mixture states with default like configuration plus lateral prefrontal cortex,
Yeah. Mhm.
and so on, and other kinds of things seem to be quite prevalent in non-minor ways.
Very
So
much.
what is your view of dynamics in general, and what is your take in terms of the relationship between the canonical static networks,
Yeah.
whichever version of the canonical one wants to adopt,
Mhm.
and what's going on really more dynamically? So that
Yeah.
this is something that I think that would be a
Yeah.
direction for the field to start thinking more broadly about these issues. And although
Yeah.
the community, as you say, is very active on it
Yeah.
doesn't feel that it has caught on.
It's just because it's complex, and any complex thing takes a while, right, to catch on. I mean, there's people who've been working on it for a long time. The canonical networks happen because the original types of analyses involved averaging across the entire time series. And on average, you'll see default mode posterior and anterior nodes connect to each other. So on average, that pops out. Whether you use independent component analysis or anything else, the average pattern pops out in those canonical systems. But as if you look, in the fine grain, like 10 seconds, 30 seconds, 60-second window, some things pop in and out, and that's where the dynamics gives you that complexity you're talking about. And people are appreciating that. There's some like methodological concerns with how you run those analyses, and that's why it's been slower for the community to sort of iron out some of those methodological issues. So, I think there's like the controversies essentially like overwhelmed us for a while. And now there's more interesting methods for looking at dynamics, and I think it's pretty widely adopted in people who do this for a living. And maybe it hasn't caught on in like the broader neuroscience quite yet, but people who do EEG will tell you, yeah, we've been doing this for 100 years, right?
Right, exactly.
So
Like,
it depends on who you ask, right?
of
So
course, the MEG folks
yeah,
will always
exactly.
be like, okay,
Yeah.
we are inherently dynamic from the
Yeah, but
get-go.
yeah, I think what you're saying though is because we get nowadays the field is a hybrid of like computer scientists, electrical engineers, a lot of signal processing experts, and then the psychology and neuroscience folks jump in. And then there's the computational model. I think the field has become much more interdisciplinary, and so we're moving faster. Like people are adopting the methods faster. We're kind of snowballing at a much more rapid rate than when I was a grad student. You just learned one thing, and you wrote a paper. But now like we're moving very quickly. So I think I'm hoping the adoption of sort of dynamic perspectives and analyses is only going to accelerate. To me, I've already seen it like accelerate a lot in 15 years.
And your take also, like, more maybe like clinically, in the clinic
Yeah,
more direction, is that these dynamic aspects might be or are important for understanding individual
100%.
differences. And
Yeah. Yeah, I think so. And that's why I want to make that clinical translation even more rapid so that people aren't getting the wrong idea. Like a psychiatrist who only has 1 day a week to look at this literature might get the wrong impression that we have these static networks and that's all there is to it, right? So we want to
I
make
see.
sure we get
Yeah,
the word
yeah,
out.
yeah, yeah.
Yeah,
Thanks so much. Yeah,
yeah. That's
yeah.
it. Yeah.
Patrick, please.
Hi. Yeah, so my work focuses on functional connectivity gradients. And I guess this depends on to what extent you think that gradients are like an intrinsic coordinate system to describe functional connectivity. But yeah, I don't know. How do you think of gradients as being helpful for resolving this like taxonomy issue? If you have these intrinsic coordinates that correspond to like functional dimensions, then does that help you sort of sidestep the rigid network boundaries and naming questions if you can decide on what the gradients mean?
Yeah, I mean, gradients just make things worse, right? Or they make it more complicated, right? And in a good way. And I think like all of these tools and approaches reveal things about the brain that show that, okay, we've been oversimplifying. We've been assuming that, there's these static things, and the more you look at it, I mean, I think that parcellations give you these hard boundaries, and people like that because it's easier to say, I'm going to work with these 200 parcels, right? It's a simplification we use, it's a data reduction essentially, right? And I think we like data reduction for the computational reasons, but every time we do it, we lose something, right? And maybe we create hard boundaries where there aren't hard boundaries. That's what the argument would be. And I'm very sympathetic to that. I'm not saying that everything we've done is justified. I think everything we've done in the field is like for a reason, like simplification, data reduction, ease of communication. I don't whether or not there are networks in the brain and whether they actually exist in some ontological way. I think we've done things like oversimplified so that we can communicate results, or oversimplified so that our server doesn't crash, right? Like we've done all these things because it's the best we can do as scientists. But the questions of like, have we done the most accurate thing for describing brain function or brain dynamics? To me, like it's an open question. Like maybe we're getting closer and closer with all of these theories, with gradients, with these different ways of thinking about it. But I think the question of what is the field doing and what's good for scientific progress is different from like, how close are we to the truth? I think to me those are two different questions. So it's hard to get the field to like move in a certain direction, right? Like it's hard to get paradigm shifts because for a while nobody would publish. I'm old enough to know that you couldn't publish a resting state fMRI paper in 2002 or something. Like nobody believed it, right? No, everyone thought it was noise and nonsense. And it was only after like a lot of methodological innovations and a lot of crosstalk with other, confirmatory kinds of analyses, work in animal models, validating that it's really neuronal stuff driving, those correlations. So I think like we've made a lot of progress towards, and now we have almost the opposite problem of like everyone believes everything you see, in resting state fMRI data without going back to, confirm the physiology. So I think these are just kind of like trends in the field, and like, we were trying to get closer and closer to some truth. And the idea that there's 6 networks, I think, is absurd. It's more of just like this has helped us as a shortcut for moving forward and thinking about findings in some. It's like for us to understand, not that we've found the answer, but it helps us get closer. So
Yeah,
the bad answer, I know, but I'm just saying I think we're just moving along, but we're not there yet.
Yeah. And I wasn't I totally agree that it's necessary to have these like almost shorthands or like refer to, we have to name things and we have to group things. And it's interesting that you say that gradients make things more complicated. To me, it's like in my head, they make things simpler because basically like networks, like the canonical 7 networks or whatever you choose, are like very well partitioned in a 3-dimensional gradient space. And so it doesn't really matter where you draw boundaries precisely. You have sort of, if you imagine like brain regions in this embedding space, then one direction corresponds to being more default modi, and one direction corresponds to being more visual or more somatosensory, and one corresponds to being more frontoparietal control or whatever. And so, yeah, I don't know. I guess that's not helpful. And I'm still using the network names to talk about those directions.
Well, that's what I mean. It's great. I think it's good to introduce gradients to give that perspective and say like, there's these parcellations, and then there, it's a way of reminding us that we could be wrong about everything, and so we have to like keep thinking of new analogies for brain, organization until we get it right, or until we get closer.
Yeah, awesome. Thank you.
Thanks.
Yeah, Henry, before I get to you, 1 second, I just want to remind people that if you are you don't have a good microphone, or you prefer to, you just write your question on the chat box, the messages there, and I can read them to Lucina. So feel free to use that as well. But Henry, please go ahead.
Yeah, thanks for your talk and organizing this. I work with intracranial electrophysiology data. And one of my big frustrations is that I have a really hard time understanding the brain in terms of electrophysiology vis-a-vis fMRI brain networks, because that's where so much of the science is. LFP data, which is what I work with, is a closer proxy of neural activity than the bold signal, right? And there are some ways in which these correlate with one another. So for instance, in task contexts, high gamma activity, so like high frequency activity, correlates pretty well with bold activation. But when you start to try to create, to sort of reproduce the fMRI brain networks at rest, this generally breaks down. And some studies will claim to do so, but they're relatively undersampled. And I don't replicate some of the same things. My question is, do you think we need to have a better biological understanding of what fMRI networks are before we can ever get to this taxonomy? Or do you think we can sort of sidestep this whole process? Sorry, it's kind of long-winded.
Oh, no, this is a great question. And I think that work that you do is critically important for, getting us to that point, like you said, closer to the actual, neural signals. I think a lot of work is actually being done. The review I mentioned, the 2025 Nature with Bharat, if you look in that one, it talks a lot about the neurophysiological origins of bold and this and that, and the animal work, right? The sort of, mice, the rodent fMRI, where you can actually do calcium imaging and all that. So a lot of that is being done. It's ongoing, and it's very, complementary to the human imaging work. So that's great. But I think people often forget that the stuff we talk about with resting state fMRI is kind of like a 0.01 to 0.1 hertz frequency signal. So it's this really low frequency thing. And you're talking about, gamma and these high frequency things. And so it's not surprising to me that they're not sort of one-to-one, this one and the same, right? They may be nested sort of frequencies in there, but like I would be surprised if they were exactly the same, right? Given we're looking at these different frequencies of oscillations. So I think that's a problem where we do need to get more of this sort of EEG, MEG, the people who have been thinking along these, faster and slower time scales for a while. Because fMRI, we sort of sweep this under the rug, but it's a slow signal, right? It's one of the slower ones, and it's useful, it's easy to use, and I think that's why you see much more literature using that tool than you do. Like the work you do is more harder to come across that data, right? Or the animal work is slower. And so I think just because there's a lot of fMRI papers doesn't mean it's like, the be all end all and like, how do we organize the brain? I think it's just, this is how the field evolves. Like you see a lot of work and everyone assumes like, well, these are the networks, this is what we're calling them, and I'm trying to get us to sort of pause and say, well, is this the best way to move forward? Is this the closest thing to the truth? And I think, adding in these data points from, these different parallel fields is going to be where we need to go next. It's just been so hard to do. Like it was hard enough to get a bunch of fMRI people to sit down and talk, and we never resolved anything, even amongst us. Imagine bringing in the electrophys people, imagine bringing in the rodent person. I want to do that. I'm just tired at this point from years of trying. Thanks though.
Yeah, no, it's interesting because there's I have a little bit of interaction with rodent people now, as a real outsider, double outsider in a sense. And they're very, because they're a smaller community, they are very close, and they talk a lot to each other, and they're very collaborative and whatnot. But it's very also insulated from human, so the best practices also in some parts might be better than some of the things that we definitely done from what we did early on in human networks, because they have quite a lot of work that people did, right? You've done and other people have done over the many years, but there's so little interaction with the human part. It's something that I find very disconcerting that. But getting back to your point too, one thing that you might be interested in looking at, and I think that's possibly a fruitful direction in one sense, although the rodent brain is very different, obviously. There is more and more wide field calcium indicators that are being used, and at least for more cortical arrangement, more cortical organization, one can have a pretty decent coverage of cortex there with calcium imaging, right? So it's not 3D like in fMRI that you have the whole brain. You could look at subcortex and so on, which is often also ignored, but that's a different issue. But in calcium imaging is a literature that is growing quite fast and helping to understand the aspects that might be tied to the blood oxygenation nature of blood, of bold, and neurofiring itself, neural processes themselves, right?
No, they're good points. I guess just one more thing to add to the sort of very concrete way to say in which, like, sometimes I get confused. So, I work with intracranial data. We have lots of insula coverage. We have lots of cingulate coverage.
Mm-hmm.
And I don't see per se anything in the electrophysiology data. Even when you sort of do these nested sort of frequency analyses, you take the high gamma activity and then filter it to look at it on a very slow time scale. You don't necessarily see something that jumps out as really being. Now, it's not, this is also like, we're talking about me and what I've done in a couple of months. There's really more to look at. There's a study out there. There's a study out there that tries to do this, and I don't see the same results. But it's also not that surprising, because a lot of intracranial data is looking at very small samples, and it matters too exactly where you sample in these different regions. Yeah. So that's sort of the dominant form of brain organization when you just look at data is like oscillations, and that has very clear patterns that are mostly based on temporal lobe, or the lobe of the brain, rather than the some sort of distal connection. So anyways, I'll just stop there.
Oh, that's great. I need to think about that. I'm going to get in touch with you, because now my mind is blown.
Yeah, no, that's actually really important, and I think that the field really needs to look into that very carefully. I don't know the literature more broadly, so how much of that work already has looked into these issues that you're indicating, right? Because there's a lot of studies that are with a lot broader coverage that are coming up in the last year or two. The coverage is expanding, so there's a lot of work that's going to really inform these networks that have been studied with fMRI.
Yeah, really fascinating. Thanks for sharing.
Lucina, there is a somewhat long comment here by Leonardo. I don't know if you can take a peek and say a few words of what you think. It says it's a comment from a linguist's point of view.
Are you still on the call,
Leonardo, I don't.
I'm here. I'm sorry, I'm at a cafe, so there's a little noise around me.
No problem. Yeah, did you want to read anything out loud or just
Do you want me
Yeah.
to read it for you?
No, no, if you want, I can read it. But as I said, there is some noise around me, so I'm sorry if it disrupts my. So anyway,
Yeah, let me
Mhm.
yeah, okay.
let me read it because I think there's some background noise as you said. Yeah. Okay. Okay, so I'll read it for you.
Mhm.
So,
All right.
sorry, Leonardo says that it's very difficult for him to imagine a world where brain networks do not exist, even if we can't agree on one yet. For instance, let's say even in terms of the language processing networks, there seems to be at least some steps that are necessary in order to process language. And even though brain processing is more complex than one region does one thing and the other does another, some form of organization seems to be necessary. So even if you speak in terms of parallel processing occurring everywhere, it would be specific mental operations occurring everywhere, somewhere. Even if we acknowledge a certain level of flexibility in processing, otherwise we would, I'm having a little bit of difficulty parsing the text here. Otherwise, we would also need to completely disregard the contributions of lesion studies. So I guess what Leonardo is saying in a way is that he seems to be pretty convinced that networks exist because there must be some principle by which regions, a functional organizational level above region has to be instituted so that there can be something that is multi-region to accomplish something based on the specialization of regions given lesion data, right? So there's functional specializations in some sense that we can't deny. So if there's functional specialization, it can't be isolated. So there must be networks. I don't know if I'm reading this right, but that's maybe one way of reading this comment. So I'm wondering if you have
I would never say there's no such thing as networks, even though we did have that discussion for a few weeks in our OneNet group. But I think the idea is we wanted to move beyond the one brain region does one thing. So okay, it's a bunch of regions that does various things, and we call those networks because they seem to co-activate or they seem to be related to each other. So then that's great. There's networks, but then the question is well, what constitutes a network? What a bunch of brain regions doing something together. What are the and then of course what do we call them, which is the problem that I've been harping on. So it's not so much like, oh yeah, there's no such thing as networks. It's more that we've been, I think, a bit loose and imprecise in our usage of these terms over the years, and that's fine to some extent when in the discovery phase of anything. I think once we started using this language, we, like I said, there was some consistency, like people seem to call the default mode network consistently, even though sometimes anterior, sometimes posterior, sometimes subdivision. It seems like there we've had some good, basically kind of robustness to the use of the term. But then there's these others that I mentioned that it kind of get us all over the place a little bit. So yeah, I totally agree. I'm not saying we have to do away with the notion of networks, but more get a little bit better about what it is we mean when we say such and such network. And just kind of maybe be honest about the limitations of what we can infer with our current scheming or current label schemes and things like that.
Yeah, so Leonardo, I don't know if I did justice to your question. I apologize if I distorted it in some way, but I don't know if you want to correct anything. I'm either on that seems fine. Okay, all right. Thanks for your point. Yeah. Okay, I'm gonna turn now to Christy. Please.
Hello. Thank you for your talk. Actually, I just started my master's degree and I do work with some level of fMRI data, so this was a good refresher for me in terms of networks. So I'm sort of thinking about, we're talking a lot about the functional data, obviously. We've mentioned anatomical data, We've mentioned other forms of data that are more closely correlated with actual neurosignal. Now I'm thinking in terms of the type of task batteries that we might be looking at when we're defining these networks, because it very much, like you mentioned, it very much be the case. It very likely is the case, actually, that several of similar tasks activate the same networks or the same set of networks, right? And when I think about the classification of those task batteries, I get overwhelmed because how can we possibly come up with enough things that the human body and mind is capable of, enough so that we are able to parcelate these networks?
Yeah.
Yeah. So I was
Yeah.
just wondering your thoughts on that matter.
No, it's a great question. And if you do a lot of sort of meta-analytic work with fMRI data, you'll notice that some domains are overrepresented. You know, people do a lot of tasks with working memory or something like that, and maybe fewer on some like lesser, I don't know, sensory processing. Maybe there's fewer tasks. So even like if you look across, hundreds and hundreds of fMRI data sets, the problem you'll find is that some things are overrepresented, some tasks are underrepresented. So you can never we'd have to write a big grant to do like every task essentially, or every conceivable task, and then get data from hundreds of subjects to really have the breadth of the fMRI task activation maps to do the kind of meta-analysis that would really inform these questions. Instead, what we have is kind of we pick up the pieces that are already out there. So you have like in the Human Connectome project, 7 different tasks, tapping 7 different domains, but they don't get every domain. So I think that that's a kind of a bigger problem, is that like no one will fund the study that would actually be needed to be done to get a definitive like sampling of most possible tasks in the world.
Right.
Yeah.
But well, in the same vein, do you think that there might also be motive for standardizing this battery, just as we're standardizing the networks?
Yeah, I mean, 100%. I mean, all of this stuff is like easier said than done. Like it would be
Right.
great. Yeah, it'd be great if we could come up. Well, in fact, that's what a lot of these large data sharing initiatives do. They kind of standardize. They say, okay, we're going to collect 7 tasks from several hundred people in the Human Connectome project. We only collect 3 tasks from 10,000 people in the ABCD study. So, we do that for these big population neuroscience data sets. And even still, it's hard to decide which ones you include. If you're only going to include 3, which ones, and then you have to get money, and getting money is harder and harder and harder now.
Yeah.
So yeah, I mean, all of these things are like, if we had infinite budgets and infinite resources, we could obviously do a better job with our.
Yeah. It's
Yeah.
always about the money at the end of the
Yeah.
day.
It's always about the money. Yeah.
Yeah. And I just have one more thing to say, unless if someone would like to jump
No,
in.
no, go ahead.
Oh, okay. So my work essentially right now, my project is looking like inhibitory RTMS to a certain brain region. And I'm basically creating what is a virtual lesion, which is why what Leonardo said was very interesting to me in terms of lesions showing us that the brain does follow some type of hierarchy. And I'm thinking that because when we're standardizing these networks, then the groundwork that goes into it is understanding the very dynamic and often fluctuating interactions between these networks, right? So we'd have to gain a better resolution of understanding. And with a better resolution of understanding, I'm thinking that perhaps one of the consequences of the standardization would be that we would be able to better predict the consequences of certain lesions or certain knockout areas, right? So do you think that might be applicable in the future in terms of a medical sense?
Yeah, I mean, exactly right. I think even with the incomplete understanding we have today, people are already using RTMS to treat depression. And
Yeah.
part of that, the reason it works is because we know like the dorsolateral prefrontal cortex is connected in some way to regions of the genual ACC that are involved in depression. So the network neuroscience is actually informing the treatment with TMS for depression. And I think like
Yeah.
knowing that is like, even though we have incomplete knowledge of these networks and what to call them and where they are, people are already applying it in the clinic, right? So I think that's why we have to work even harder and faster to get our story straight, because whether we like it or not, it's already being translated, and it to some level of success. But the better we can do in our like delineation and our anatomical specificity, the better the clinicians can do with targeting treatment and things like that. So that's why I think it's like even more impaired. Like it's fun for basic science, but it's like critical for us to get it right.
Yeah.
So
that's a really good point. I'll definitely have to bring that into my my own thesis research, the idea of like different networks and how they might apply to TMS.
Yeah.
Thanks. Thank you so much,
Well,
Lucina.
I can send you, there's a lot of papers now using TMS targeting for different clinical conditions. So it's already being used. Yeah.
I would really appreciate it. I'm doing
Yeah.
my literature search right now. I could use all the papers I can
Yeah,
get.
Mike Fox at Harvard does a lot of this work and has a lot of good reviews as well of different TMS targets for different clinical applications. There's a lot of, I mean, there's so much translational work here that's why I think it's like super important for us to you know, at least get our story straight for
Yeah,
for export.
absolutely.
Like
Thank you so much. Yeah.
Sure.
Thank you.
All right. So any final questions while we have Lucina here with us?
Yeah, I have.
Okay, great.
I have a quick question.
Okay, go ahead. Go ahead. I can't
Um,
see where you are, but just please go ahead.
Okay, yeah. So these networks that we construct in fMRI are typically constructed using a fairly simple measure, Pearson's R. I'm wondering just what you think about, this only considers linear associations between regions.
Right.
I'm wondering what you think about other measures that consider nonlinear associations, or even things like representational connectivity, which looks at the representational content of regions and considers how those are similar across networks. And I don't know if these other forms of connectivity produce the same networks as Pearson's R, if we get different results across these different types of measures. Yes, I'm just wondering if you could speak on that.
Yeah, I mean, it's a great question. I think, I mean, Pearson's is probably like the most commonly used way of pulling out some of these correlations, but also, independent component analysis is pretty widely used as well. Oh, there's a paper in the chat that someone just posted that looks relevant as well. But yeah, I think you're right. I mean, there's other ways of computing, of getting at, what you would call a network, and the representational stuff. I haven't seen as much of it, but I think you're right. I think there are groups who are working on that. And it's an empirical question. If people, run different kinds of analyses and they produce something different, then I mean, it's something to think about for sure. I mean, I think the problem is for better or for worse, we've decided that this statistical measure of correlation, for us, we're going to call it connectivity, and we're going to use that as our sort of basis for understanding interactions between brain regions, for better or for worse. But there are different ways of doing it, like you said. And so I know there's groups working on it. I'm not quite as familiar with like all the different flavors right now, of the definitions, as you mentioned. But I think like the more people do this, the more they explore the space, the better. Like, and if it contradicts what's currently out there, that's even better, because it just gives us, a new avenue to pursue. So keep doing it if that's what you're doing. Great.
Okay. Yeah, that was a great question. Any other questions? So one thing that I'm going to ask you before we leave is that if there was one thing, let's say you have the power, one thing that you would change in the field of networks and so on, right? Because not the whole universe, but yet.
Yeah.
But in that domain, if there was one thing that you could change by just saying, look, this has to be done, or this should not be done, or you get to pick.
Yeah.
If you want, you can give you even one or two. It doesn't have to be only one thing. But what would be your kind of desire to change, or to stop, or to move us in some way or another? Or what would you use that power?
Yeah, I mean, if I had the power, I would get everyone to start making sure whatever you do in your paper, make sure you add very specific anatomical descriptions of the entity that you're describing. So, some papers, you can go through the whole paper and the brain regions aren't specified. They just say default mode network or this network or that network. And like, personally, I don't even care what you call it. Just tell me what brain regions we're talking about. Are we talking about anterior insula? Are we talking about ventral medial prefrontal cortex? Because it's harder to disagree. You can still disagree about anatomy, but it's a lot harder to disagree about coordinates and about, MRI coordinates and about like, really anatomically specific descriptors of your finding. Because I think the problem is just that we've gotten carried away with, it's great to give a functional interpretation of your findings, but if that is in lieu of giving a good anatomical description, then it's hard to build on it, right? So
yeah, and I think that actually gets to the best practices of a field, right? It's almost like something that we should, in statistics, there's a lot of controversy in statistics, but there are some best practices that you report, and you state the test that you did, and you state p-values, you state this and that assumption or then whatnot. So that part, I think that it should be doable, right? If journal editors and reviewers became a little bit more mindful of that and a little pickier, saying, okay, you didn't, please bring the parcellation that you used and discuss that right away, and not in a footnotes 37, that no one sees that, only in supplementary material or something.
Partly the reason I say this is because people are doing a lot of text mining, which is great. Like, I mean, that's even how Neurosynth works. It mines text of analyses, and that's all great. But if your text doesn't include a complete description, then then the meta-analysis based on that text will never give you a really thorough understanding. These are really like nuanced points, and nobody wants to be that nitpicky. But like, I just wish we would be more descriptive in our.
No, I think that with the best practices, I think that makes a lot of sense. It really should be something that some of you who are starting to include in your papers, and if you're reviewing, to just kindly ask, please provide what is the anatomy of your parcellation and so on.
Use the network correspondence toolbox. It's a Python toolbox. It's that's what it's for. It's like to make it easy for you. You could use those figures in your paper, and that's what it's for. So I mean, that's what we're trying to do. Is just like make it easier for people to do a bunch of reporting.
That makes a lot of sense.
Yeah.
All right, Lucina.
Someone says, does that
thank
mean
you so much.
Use AI for neuroscience research? Well.
Oh, there's one more I missed. Okay.
We can't use AI because the body of work is too confusing. Well, that's a good point. Like if you ask, I haven't done this recently, but if you ask AI to summarize some literature, it's summarizing the text, right? So, it's not going to have the same understanding. It doesn't realize that there's these controversies in the field about what to call what. So, it's a good point.
Yeah. Okay. All right. Thank you so much. I think it was an awesome discussion, and thanks, everyone.
Yeah, thanks. This was fun.
All right.
All right.
Bye, everyone. Bye. Take care.